It gives me absolute immense honour and pleasure to introduce our next keynote speaker, who in the mid-60s, when he was finishing his own PhD and starting his own lab, after mapping the catecholamines within the central nervous system, embarked on what was to become a pioneering career. He embodies what we have tried to present with this conference, which is to examine biology and look and learn how or in what way we can use that knowledge in terms of regeneration. So it was in the mid-70s when he was setting his own lab up in Lund that he actually kicked very hard against a dogma at that time, which was the CNS was hardwired and would not change under any condition, whether it be disease, whether it be age, or whether it be trauma. He believed, and his group studied the possibility that actually the brain was plastic. And I think Anders was one of the first people even to say that the brain was plastic. Jump about a decade and a half further in my own career, when I was in St. George's Hospital Library, reading one of his first papers on a group by Anders Bjorklund and Ulf Stenevy, who began to examine the possibility of taking immature tissue and placing it into an adult diseased or damaged brain to see whether it could reconstruct that environment. In the 80s, what became very clear from that pioneering work, that regeneration within the brain was possible, was feasible. And there was an amazing story which made the BBC here, which was the frozen addicts, which that story itself is amazing, as in a collaboration between yourself and Langston, who got stuck, I think it was in a ditch outside Belgium, and decided that a group of drug addicts who'd actually used a designer drug, heroin, which contained a toxin because it had been made wrong, resulted in basically young people becoming Parkinsonian. 3 of those patients travelled to Lund with Hagen, Widner, and Ollie Lindvall and Anders, who then operated on those individuals who were totally frozen. At the time, because they were drug addicts, it was thought that they were in some way catatonic schizophrenics, but in fact, they were Parkinsonian. But L-DOPA only gave them minimal relief for a short period, and some of them didn't work at all. But they received some of the first fetal transplants to relieve those conditions, and in fact, 3 of those, Connie, Juanita, and George, thankfully now are able, even now, to be back and able, and thankfully they've not gone back down the path which took them into drug use. But in 1990, that procedure was also used by Ollie Lindvall, who again was a student of Anders, to look at the use of fetal transplantation for Parkinson's disease, which has led to the use of possibly using human embryonic pluripotent cells to actually do a similar thing. So not only within the Parkinson's area, regenerative biology, mapping CNS anatomy, also within learning and memory, and a very active collaboration with Fred Gage, who again was a postdoc of Anders. So as you can see, he's a major hero of mine. I totally blame him for my career, which is not in CNS, well, it is in CNS, it's in the eye. So my career within regenerative medicine, I totally blame on reading those papers back in 1984 in St. George's Hospital. So it gives me great pleasure to introduce our keynote speaker, Professor Anders Bjorklund.
Well, thank you. Thank you very much. That was almost too much to cope with. I don't know. Perhaps I should cancel my talk. Perhaps I should cancel my talk. But, so I will not be telling about the sort of the early history of cell transplantation when we did our experimental work in rodents. But our focus on the clinical program that Pete said, we initiated in Lund with Ole Lindvall as the leading neurologist in the 1980s. So this is now about 30 years ago when we asked for permission to use fetal tissue from aborted fetuses to be used for trials in Parkinson patients. And that was by itself an interesting exercise because it didn't fit to the transplantation law we had, it didn't fit to the abortion law. So we had to go to our Swedish Institute of Medicine and ask their ethical committee to tell us what the ethical rules should be. And that eventually it was turned into a separate law by the parliament in Sweden, and then we could go ahead. So that was an interesting exercise. Well, this is about Parkinson's disease. I guess you all know that this is a disease of the motor system in particular, and the motor symptoms in the disease are related to the loss of dopamine neurons in the midbrain. And this is the core of the pathology. We know that many other things happen in the brain as the disease progresses, but this is what presenting the disease in idiopathic Parkinson's. It's the dopamine defect that's leading the symptomatology. And this is progressive over time and leads to a defect in initiation or movement. This is a very simplified diagram of how we execute our motor output. We command it from the cortex, goes through the striatum to the thalamus, and results in a motor response. The striatum is critical for initiation of movement, and this in turn is regulated by the dopamine neurons in the midbrain. What's interesting is that dopamine neurons themselves are not linked into the execution pathway. The execution pathway is red here. So it's organized sideways and is setting the threshold for activation of the striatum. So it's a bit like a clutch in a motor. The motor is intact and the clutch is there to put the motor in gear. So when dopamine neurons are missing, the patient is experiencing a resistance to the initiation of movement, and it becomes slow and cumbersome. What's the idea behind using dopamine neurons to reactivate the motor system is quite simple, and it's also, I think, supported by the fact that L-DOPA therapy works very well in the early stages of the disease. So restoring dopamine neurotransmission in the striatum, even by injecting L-DOPA or taking L-DOPA by oral tablets, is sufficient to reactivate the receptors and restore, reduce the threshold for initiation of movement and restore motor function. This can be monitored by positron emission tomography. And when we started in the 80s, this was a very welcome and very important additional tool when we did the early trials, because here we have had already at the time, and it remains the best tool still, to monitor dopamine function in the striatum. So what fluorodopa does is to enter the brain, be converted into fluorodopamine, and fluorodopamine is stored in the dopamine terminals in the striatum. And it's the stored fluorodopamine that's being visualized here. And you can see that this is from the Hammersmith unit in London. You can see that over time, as the nigrostriatal dopamine neurons die, there is a gradual loss of dopamine synthesis in the striatum. And in symptomatic individuals, the putamen in particular, which is the caudal part here, is much reduced. With dopamine neuron transplants, and these are then fetal dopamine neurons, or it's actually dissected ventral midbrain that is dissected so as to contain the neuroblasts that are undergoing their last cell division or becoming post-mitotic. It has to be at that stage of development, and that is around 6.5 to 9 weeks of gestation in humans. That piece of tissue is dissected and turned into a cell suspension, and this is then injected into, in most cases, just into the putamen. We have, in order to get enough cells, we have to pool from several donors, and in our trials it's between 4 and 6 donors that have been transplanted. And here you can see that, monitored by fluorodopa, that there is a new signal growing up. Here it's at 12 months. It continues, as I'll show, over at least 3 years. And you can see here the needle tracks. There are 3 needle tracks on each side in this brain. And you can see the signal is growing up around. It's not the cells themselves that give the signal, it's the outgrowing axons. And that takes time. So what about the outcome? I'll show an example. This is from our colleagues in Canada. This is in what's called practical or defined off, which means that the patient hasn't taken any medication since the last day. You can see that this is a typical Parkinson symptoms with short gait and stiff posture, no arm swings. And here is what happens after, in this case, 12 months, and the time course of changes, I'll illustrate that, can vary between patients. And you can see here in the pre-op, when she's turning, she has this typical freezing. So the Lund transplantation program has comprised 18 patients, and some of them have been these MPTP patients that Pete mentioned. The rest are idiopathic patients. Started in 1987. The transplants are derived from 3 to 5. I said wrong. We have actually taken 3, some cases also 3 to 5 per site. Initially, they were done unilaterally for safety, and I'll get back to that. And some of these patients have been followed by this fluorodopa PET imaging for up to 18 years. And what's been particularly interesting for us is that over the last few years, 4 of these patients have died for other reasons and come to autopsy, and we've been able to look into their brains at stages between 12 and up to 24 years after transplantation. These transplants can give, in about 1/3 of the cases, significant improvement in clinically that is maintained for a long time. These are 2 patients that have been followed by Neil Quinn at Queen Square in London that were at this point when this was put together in 2010, they had their transplants 13 and 16 years. And you can see that over that time period, the transplants have grown, as indicated by the fluorodopa curve, the yellow. And you can see that it's very gradual. In this case here, patient 7, you can say that it increased up to about 4 years. And here, I think this is also 4 years. The reason why it's so slow is that this is reflecting the re-innervation of the surrounding target. And the human dopamine neurons mature slowly, and they grow slowly. It's a very marked difference between rodent cells that accomplish their outgrowth within about 3 months, 3-4 months perhaps, and the human cells that grow for years. We think this reflects the development of the human brain, which is protracted over years. And the nigrostriatal or the basal ganglia are also continuing to expand over that period. Then you can see the recovery of the motor function. Well, I should also indicate that in both these cases, the fluorodopa signal now is within the normal range. Parallel to that, you see the improvement in motor function as monitored by what's called the UPDRS scores. It's a scoring scale that clinicians use to monitor the symptoms. And the higher the score, the worse the patient is. So reduced scores is an improvement. And you can see that over the 16 years, this patient has gone from something around 40, which is a severe case. This is in practical off, down to about 12, 15, which is similar to an early stage patient. In this case, and you can see that the improvement has continued for at least up to 3 years, but it may also be an improvement. Somewhere around that time, they stopped the dopamine medication, both of them. And in this case, you can see that it goes from perhaps somewhere around 25, 30 initially, a little less severe than the other one, and it's down around 10. So in both these cases, it's a sustained improvement of a clinically highly meaningful level of improvement. Now, I mentioned that this is seen only in subportions of patients. Roger Bocker in Cambridge made an effort to get data from 4 different centers that had been doing transplantation over the years. Apart from us in Lund, it's the Halifax, the Canadian, and then the 2 US groups that were sponsored by the National Institutes of Health. And this is the scatter plot where the improvement in UPDRS is plotted at the latest time point. The American patients have not been followed very long, whereas the others have. If you put a line at 30% improvement, which is the dashed line here, as a level of improvement that would be significant, that is a magnitude of improvement that goes beyond what could be expected from placebo effects or from fluctuations in the condition, you can see that 40% of these 65 cases fall within that range. So it fits to the idea that about 1/3 of the patients can have been benefiting from the surgery. Now there is one interesting patient here, over here, which is patient 4. As you can see, this is the patient that has been followed longest, and it was the 4th patient in the Lund series. The first 2, they were operated unilaterally with too little tissue and an instrument that was to thicken a caliber to secure that the cell material was retained at the right place. So they have not really given any information. But the following 2, patient 3 and 4, were the first 2 that showed interesting changes. And this patient 4, he was diagnosed in 1980 at the age of 50. 9 years later, he received a unilateral transplant from 6 to 7 week donors, from 4 such donors in the putamen. And over the next 3 years, it showed gradual and marked clinical improvement to the point that he could stop all medication at 32 months. The first 5 patients beyond the 2 patient 1 and 2, they were operated unilaterally first for safety reasons in order to follow them sufficiently long time. And then they were offered to have a second transplant on the other side. 4 of the 5 agreed, but this one, patient 4, he felt he was doing so well with just 1 transplant, so he didn't want to a second surgery. Now, that turned out to be very interesting because this means that he remained having a transplant on one side and the other side as an internal control. So Olle Lindvall, who was actively monitoring these patients, he followed this patient closely over 10 years. And I'll get back to that. And then in September 2013, he died at the age of 83. At that point, he had developed dementia. The last few years, he had gradually declined and became demented, indicating that these transplants may do good things on the motor symptoms, but of course the disease will progress and other symptoms that may appear like dementia will progress unaffected. Now, this patient has resulted in 2 publications. One in '92, where patient 3 and 4 were reported over a 3-year observation period. And then in 1999, Paola Piccini and Olle made a separate investigation at 10 years after transplantation in a paper that turned out to be very interesting. So this is the development of his clinical condition. So here in yellow is UPDRS motor score over 10 years. You can see that, again, the improvement takes about 3 years to become prominent. And then he's remained essentially on the same level for these 10 years. At a level which is, this is at 32 months he could stop dopa treatment, so he's without medication. And you can see that his daily timing off, which was, he was off 2/3 of the day prior, even despite optimal medication. And with the transplant, he's on all the time. And here you can see the other interesting curve. The non-grafted putamen will continue to decline in fluorodopa uptake, so indicating the progressive loss of dopamine neurons, whereas the grafted stratum, there is this slow recovery over about 3 years into the normal range, which you see here. So his fluorodopa dopamine production in the stratum is back to normal. At this 10-year time point, Paola Piccini made a test for dopamine release that is based on the use of raclopride for dopamine receptor displacement. Here is, well, I should, by the way, just show the increase in fluorodopa signal there on the right-hand side, and then the continued loss of signal here. And here is, now before I move on to the raclopride, I should tell about this, that the brain came to autopsy. This was in 2013. We have Olle Lindvall here, the neurosurgeon Stig Renkrona, and then Patrick Brandin's colleague, Yayi Lee, who is handling the analysis of the brain. And missing here is Håkan Widner and Patrick Brandin, who wasn't around at this time. The most striking is how well the transplant has survived over these 24 years. This is in the Rostow graft site in the putamen. This is the caudal graft site in the putamen. You can see the transplant in the middle with Th positive neurons, and then the reinnervation that is covering the entire putamen. Interestingly, the innervation is precisely confined to the area that should be innervated. You can look here, for example, the border to the globus pallidus down here. You can see the innervation expands up to the border and then stops. And this is precisely what we see in animals, that the innervation that is generated by these transplanted dopamine neurons is precisely matching the normal innervation in animals that are denervated prior to transplantation. This is the control side. You can see that fitting to the fluoridopa uptake signal, it's completely denervated. So the putamen in this case, he's had this disease then in, what is this now? Must be, he was transplanted in 1980, so it's 35 years from diagnosis. Here is an age-matched non-Parkinson brain where you see the innervation is still good, and the innervation by TH. At higher magnification, and this is in dog field microscopy now, you can see that there is a very nice terminal network with the typical varicosities on the terminal branches that are characteristic for the dopamine innervation. This is on the non-grafted side. The only thing you can find in the putamen are these stumps of pre-terminal axons that end bluntly as if they are truncated. And there is virtually no branches that would look like terminals. Then we've done a fiber counting with a special axon counting technique. Here is the level in the edge-matched control in the direction medial and lateral over the transplant. You can see that close to the transplant on the medial side, there is an innervation density that matches what's seen in the normal case. And on the lateral side, it's more like a gradient, but still significant innervation throughout the putamen. And the control side is essentially zero. It's well known since a few years back that brains from transplanted patients that have survived more than 10 years, one can start to see Lewy body pathology in some of the dopamine neurons. That's been a very interesting observation. You may have heard of it. It's one of the arguments that is quoted in support of the idea of spread of alpha-synuclein pathology, or the prion-like spread that would go from the host brain to the grafted side. These are the kind of Lewy bodies that are seen with alpha-synuclein straining and ubiquitin. The vast majority of the neurons look healthy and normal. These are 3 graphs that we have analyzed in Lund. You can see that up to 10 years, there is really nothing to see. And then at 12 years, it's about 2%, 16 years, about 5%. And in this 24-year graph, he has Lewy body-like or alpha-synuclein inclusions in about 12.5% of the neurons. This suggests that the addition of pathology in neurons is around 1% per year, which is amazingly slow. It starts after 10 years, and then each year there seems to be another 1% added. Why it happens and why it's so slow is something to consider, and it's very actively discussed. Now I come to what I mentioned, that at this 10-year point, Paola Piccini and Olle Lindvall made an analysis of dopamine release from the transplants. That's become quite interesting. This is a technique where you use reclopride, labeled reclopride, that's injected at low dose and will compete with the endogenous occupancy of released dopamine at the terminals, at the receptors. So it's receptor occupancy in an inverse manner. The more the raclopride binds, the lower the endogenous release of dopamine is. In order to trigger release, the patient is given an intravenous injection of a small amount of amphetamine, and then this is combined with C11 raclopride tracer. This is what it looks like in a normal subject. In baseline at placebo, there is a certain level of binding, and when you give the amphetamine, which is methamphetamine in this case, this is reduced because there is now displacement of the raclopride by the endogenous released. And this is what is seen in the unilaterally transplanted patient. The transplant is here on this side, and you can see that there is already at baseline a difference in raclopride binding between the two sides. And when amphetamine is given on the on the non-grafted side, there is no change. There is no dopamine release. Whereas on the grafted side, there is a reduction of raclopride binding, indicating dopamine release. And this is the quantification of that. This is in the baseline condition, where you can see in the grafted putamen that this denervated area has a higher baseline binding level, and in the non-grafted side, whereas the grafted side is significantly lower, suggesting that there is a baseline release. And now when you give amphetamine, this reduction in raclopride binding that's indicated here, which is about 25%, is markedly reduced in the presence of the grafts, indicating that amphetamine is further triggering release of dopamine. So we think this shows that it's an interesting use of the fact that the patient has transplant on just one side. So what are the challenges then for further development of this technique? Well, we think that there is a lot of observations that collectively support the idea that dopamine neuron transplants actually can work. There is long-term survival in the brains of PD patients. We've seen it up to 24 years. There is an efficient reinnervation of the dopamine denervated stratum linked to a restoration of dopamine synthesis and release. And there are a number of patients where there has been a significant long-lasting clinical improvement with a development and magnitude of the improvement that excludes that it can be placebo. But the results are far too variable. I mean, one couldn't use a technique like this when the success rate is so low. We need to understand why. We need to also work on some kind of standardization of the preparation of the cells. And in that perspective, fetal tissue is, of course, unsatisfactory to use. It's very difficult to standardize. It's difficult to get enough tissue, can be contaminated. And in addition, of course, there is ethical concerns and ethical problems around use of fetal tissue. So in the long run, which Malin Permer will talk about after lunch, there is a need to move towards stem cell derived cells that can be standardized and quality controlled. At this stage, Roger Barker entered the scene. He arranged over a few years a series of workshops in London that were sponsored by Parkinson's UK. And the idea was to invite all active investigators that have been involved in performing these clinical trials with fetal tissue, and then do a joint screening of the results and discussion of the experiences from these trials, with the goal to optimize the protocol and also optimize patient selection, tissue preparation. This has now resulted in a European consortium that's called TransEuro, that's funded by a Framework 7 grant. It involves 5 centers in UK and in Sweden. And the main idea is to pave the way for a stem cell trial that could possibly be designed as a parallel arm to the fetal trial test. The idea is that today what we have access to and what we know in principle can work is fetal tissue. But we need to move gradually towards stem cell trials. So what are the factors that affect clinical outcome? Why is it variable? One that is of key importance is incomplete immunosuppression. And I mentioned that there were 2 NIH-sponsored trials performed in New York, where they did, in one trial, no immune suppression, in the other one, short-term cyclosporin-only immune suppression without any assessment of compliance. So essentially 2 trials with no or limited immune suppression. Why is that bad? Well, allogeneic tissue that is from immunologically different individuals, they are to some extent protected in the brain, but the protection is not complete. There is a risk that the immune system will see the transplant, and if immunization takes place, this is going to induce an immune or inflammatory response. And it's the delayed response that is most critical here. It doesn't happen if you transplant a kidney or so, the rejection will take place pretty quickly. Here it will come creeping and slowly developing, and will compromise both survival and axon outgrowth as well as function, which means that immunosuppression, and the experience is that it has to be complete. In Lund it's been triple immune suppression, and it has to be maintained for at least 12 months. In one of the NIH trials, the improvement curve looks like this. Here is the placebo group. There is no placebo effect, which is interesting. This is a blinded study. That was the main point, actually, of the NIH trials. And here is what you see. And if you compare that with 2 of the small cohorts in the Lund program, you can see that the improvement over the first 6 months is as expected. And at 6 months, this cyclosporin treatment was terminated. Single low dose cyclosporin. And at that point, the improvement stopped, and then it reverts back. This is a curve that is unexpected, but it would fit very well with a delayed immune response. And Jeff Cordover, who's been doing much of the analysis of the brains that have come to autopsy, he has observed that in these transplants, and I think this is now 18 months after transplantation in this case, there is a marked microglial activation that's sustained over a long time. And you can see that this is the one of the placebo control cases, and this is the grafted case. This kind of response is known from experimental work that this is going to compromise dopamine neuron survival. This is from the Stockholm group, Lars Olsson and collaborators, some years back, where they looked at transplanted between rats that are different both on class 1 and class 2 markers, immunologically different. And in this case, they triggered an immune activation by taking lymphocytes from the lymph nodes of animals that were immunized with the same with tissue from the same donors. And then after, I think it's 8 weeks, they injected the lymphocytes. And you can see that this is now generating an immune response that is classified according to a schedule they have. And you can see that this is correlated with a reduction in TH, survival of TH neurons by at least 50%. And this is the curve of functional effect. You can see that here is the graph group that improves over 8 weeks, and this is the rotation response. And in non-manipulated animals, this will remain stable over time. So in rodent experiments, allografts can survive well. This is the active immunization with the lymphocytes. You can see that this induces an inflammatory response that makes the function revert to normal. And this kind of curve, illustrated by the yellow here, is very similar to what they observed in the New York trial. So this is the picture of what may happen with allogeneic transplants based on the animal experimental work in rats and mice, that in the short term there will be an immune reaction, an inflammatory reaction around the grafts, inevitably. This is illustrated by an upregulation of both class 2 and class 1 markers, as well as an activation of microglia. In cases where the host is not immunized, this can develop in 2 ways. One is into a healthy graft, which in rats and mice will perhaps be 6 to 8 out of 10 animals that will remain healthy, whereas the other ones will remain immune activated and then can very easily develop into a delayed immune reaction. And this is illustrated easily by looking at microglia and class 2 markers. If the animal is immunized, which happens in some cases without doing any immunization, then this will be seen as a rejection. And that will happen in a few cases in each group. So in order to avoid this in rat or mouse experiments, one will use inbred strains where the immune differences are small. Then with the inbred strains, there is very little risk for rejection. So this is then suggesting that one really has to immunosuppress. The second factor that's important is patient selection, something that both Olle Lindvall and Roger Bacher has been very interested in. There is indications from the analysis of the overall outcome of transplantation that graft efficacy may be reduced in patients with more advanced disease, suggesting that the best results are with patients that have still a good L-DOPA response and where the disease is quite pure motor disease and with little dyskinesia induced. And one aspect of this is the extent of dopamine denervation. We transplant only into the putamen, but of course the dopamine system is innervating much broader regions in the forebrain, caudate nucleus, but also ventral stratum and cortical areas. And in particular, the ventral stratum is interesting. And Paula Piccini and Olle did a retrospective analysis of 2 cohorts of patients. They took a group of patients with good outcome and another group of patients with poor outcome. And Paula then measured the extent of dopamine innervation loss in ventral stratum as assessed by fluorodopa. And these pictures now, they illustrate the magnitude of loss not on the magnitude of the signal. And you can see that in the patients with poor outcome, there is more widespread and more profound loss of innervation in ventral striatum than in the good outcome. We've done animal experiments where we have denervated the forebrain in 2 steps. We denervated the striatum first, transplanted, got a functional recovery. This is in rats. Then we have gone in and lesioned the ventral striatal input in a second surgery. And then we see that there is a significant loss of the motor function that was regained by the transplant. So it means that the total motor output is dependent not only on putamen, which is otherwise the motor part of the striatum, but on the collective function of the wider striatal area, including the ventral striatum. There is support also from David Eidelberg's study of the patients done at Columbia, and he's plotted the baseline fluorodopa uptake in the ventral rostral putamen, which is part of the ventral striatum. And there is actually a significant correlation with motor improvement also in that group of patients. So we believe that this is another, as long as we don't have enough cell material to transplant more areas, it becomes important what remains in other parts. Then the third issue I'm going to touch on is the composition of the graft tissue. And this is important in 2 ways. We first are quite convinced that there is a minimum number of dopamine neurons surviving in striatum that will be needed for a good clinical outcome. We think that this is in the range of 50 to 100,000 surviving dopamine neurons. It's of course very difficult to be sure, but it's based on what cell counts in brains that have come to autopsy. And then the other aspect is that it's only one subtype of dopamine neurons that is functional. And the background for this is this picture here. In the brain, there is 9 different dopamine cell groups. And 3 of them are down here in the ventral membrane, red. And the others are part of the diencephalon, hypothalamus, and 1 set of neurons in the olfactory bulb. It's an experience from use of stem cells that many protocols are generating Th-positive neurons. But when looking at other markers, it's clear that they, to a very limited extent, are midbrain in phenotype. The so-called forebrain protocols that generate neurons are mostly of forebrain character. They are more like the olfactory bulb interneuron type, which are GABA containing, whereas these ones do not express the GABA markers, and they critically express, for example, this marker PTX3 that's characteristic for the midbrain neurons. Now, these are of 3 subtypes. There is the A8, A9, and A10. A9 is the nigral neurons. Both these types, the nigral and VTA neuron types, that's A9 and A10, are clearly present in fetal VM transplants that are studied in rodents. And they are characterized in 2 ways. One is by the morphology. The nigral neurons are bigger and more angular, and the VTA are smaller and more rounded. And then there are markers that allow distinction, immune markers that allow a distinction between the 2. I'll get back to that. Lachlan Thompson, who is a former postdoc and now back in Australia, he made an experiment to study which neuron type is innervating the striatum and which neuron type is innervating non-striatal areas. So the VTA neurons are innervating limbic and cortical areas, including the ventral striatum. whereas the nigra neurons are innervating the straight and proper, including the caudate and putamen. So he transplanted into neonates where the innervation of both cortical, limbic, and striatal areas is prominent. And then he injected tracers into either the anteromedial frontal cortex or into the dorsolateral straight. And this is normally purely nigral innervated, and this is normally purely VTA innervated. And the tracing experiments combined with the characteristic markers show that the stratum, the labeling from stratum labeled exclusively A9 neurons characterized by this GERK2 marker and by the morphology, whereas the cortical injection labeled exclusively the A10 neurons characterized by this Calbindin marker. And you can see, if you look carefully, you can see the double labeling here. So this suggests that the transplants retain the target specificity also in the adult brain, which is interesting, that even though you inject them as a mixture, they will seek out their appropriate target. And it's particularly striking for the VTA neurons because they have to grow through the stratum in order to reach the anteromedial frontal cortex. But it means that they don't extend any terminals while they're growing into the stratum and ramify only when they reach the cortex. And conversely, the nigra neurons will reinnervate the stratum, but they will not extend axons further into the cortex. Now, in order to show that only the nigra neurons are functional, we made use of this transgenic mouse. PTX3 is expressed in the ventral midbrain, and PTX3 is required for survival of the nigra neurons, but not for the VTA neurons. In PTX3 knockout mice, the nigra neurons are formed, but they die, and only the VTA preaction is left. So we made use of this and transplanted heterozygous and homozygous VM into the stratum. Since this is a knock-in mouse, we can visualize the cells and their output by expression of GFP. So these neurons are GFP positive. And in the transplants, and this is, I think, 3 months afterwards, the grafts that have both alleles of PTX3 knocked out, they will contain only A10 neurons. There are no A9 neurons left. And this is illustrated by these markers. You see no GERK2 positive, TH positive neurons in the graft. And when they are transplanted, one can see that the innervation of the stratum is much reduced in the animals where the nigra neurons are depleted. And when looking at the functional recovery, and this is two kinds of tests, this is the drug-induced and this is the spontaneous test, you can see that the complete transplants with both types of neurons, they are compensated, whereas the ones without the nigra doesn't show any significant improvement. So the behavior recovery here is correlated with the content of VTA neurons. So how about human tissue? I want to finish by showing a few slides from a material that Malin is going to talk more about this afternoon, where we have compared fetal VM tissue with the cell-derived dopamine neurons. In this case, we have transplanted the neurons into the nigra. We think this is an interesting way to test their growth capacity when it comes to how far they can extend their axons and also their growth specificity. Interestingly, human cells, human neuroblasts transplanted into the nigra, will go axons very efficiently along the nigrostriatal pathway and the medial forebrain bundle to reinnervate areas that are normally innervated by dopamine neurons, and this is in animals where the dopamine system has been removed by a 6-hydroxydopamine lesion. And you can see that both striatum up here, as well as VTA areas like the accumbens, septum, and anteromedial frontal cortex and the perirhinal cortex over here, they are well innervated. That's to say, both nigral and VTA terminal areas are innervated by these. And if we look into the bundle, it's striking how well the axons are following the two bundles. The nigrostriatal pathway is carrying the nigral axons, and the medial forebrain bundle, the VTA axons, and they can be traced all the way up to the striatum. We've also looked at the glial component of the transplant, and this is another interesting aspect, that the young growths, we can't see any glia generated, but they are as efficient in growing axons as the older growths where we can see glia, suggesting that glial component of the transplant may not play a role. And here is the more detailed pictures of the innervation in the striatum, innervation in the accumbens, and innervation in the anteromedial frontal cortex. This is the A9 nigral target, and these are the VTA targets, suggesting that also the human cells, despite that they are transplanted into a rodent brain, will be perfectly capable of seeking out the right targets and do that over distance. And not just the targets, but also the right axonal trajectory, which suggests that there must be cues present in the adult brain. So want to end by commenting on what is now being planned. We have the Trans-Europe program, the one that Roger is coordinating. It's funded by an EU grant. And the plan is to do 20 patients that will receive fetal VM tissue, bilateral imputamen, with an optimized protocol, and designed to pave the way for a stem cell trial that hopefully will come within not too long time. Then we have the second program that's also EU sponsored, which has one aspect of the program is to generate GMP certified dopamine neurons from human embryonic stem cells. And this is a program that's led by Malin, and you'll hear more of that after lunch. And the third initiative is what's been named G-Force, to put some gravitas on the program, I guess. This is an international consortium with teams from US, it's particularly New York and California. From Japan, it's the Jun Takahashi and the Yamanaka Institute who's leading that one, as well as our European program. And the idea behind this initiative is to benefit from coordination, to try to approach hurdles and difficulties in bringing ASLs to the clinic in a joint manner, share experience and share protocols. And the spirit in the group is fantastically positive. We're all both surprised and very pleased about that. There is really a strong wish to collaborate in this way. It may mean, of course, that we can't keep our secrets to ourselves, but we will, of course, share both setbacks and possible successes, which is more important. So these are my collaborators on the preclinical side, with Malin Palmer in the lead, and Shane, who's done much of the in vivo work. And then the clinical team led by Olle Lindberg and Håkan Widner. and the neurosurgeon Stig Renkrona. So, thank you.
That was a fantastic talk. I learned a lot. Thank
Okay.
you. I was wondering, do you ever see evidence for really long-range axon growth? There's a spinal dopaminergic innervation. I'm not actually quite sure whether that disappears in human patients, but do you see re-innervation of longer tracts?
We haven't really looked carefully, I must say. With rodent cells, the axon outgrowth from the midbrain to the forebrain is more limited. We can get this to happen in mice, so mouse to mouse, where the nerve pathway is short, but we cannot get that regrowth with rat to rat. Okay. We think it's because these cells don't grow for a long enough period, whereas if we put human cells in rats, they grow very well. So I think your question should be addressed with human cells into a rat. Okay. And we know that there are also non-dopamine neurons that project. So your question could be answered in two ways. How much long projections are there for dopamine neurons? But we can also look at the non-dopamine component of the growths, how far they grow. But we don't have much information on that.
Okay, thank you.
Okay, I mean, I understand that you dissociate the cells. Do you only put neurons or do you put glia? Is that important? Yeah. Or what happens if you do minimal dissociation, put little clumps?
Well, these are questions that have been really much in focus. We don't make any purification on the material. So the composition is determined by the age of the fetus that we dissect. And we know that the dopamine neuroblasts is only between 5 and 10% of all the cells. If we do a smear and count, it's between 5 and 10. So the other 95% are non-dopamine, non-TH expressing. Some of them are probably earlier precursors that we think that there is 1 or 2 cell divisions in these and then generating more dopamine neurons. But the rest are other neuron types. We know that there are serotonin neurons, there are glutamatergic and GABAergic neurons in the mixture. And with the older transplants, older donors, we see that there is quite a bit of gliogenesis going on. So we can detect glia, and interestingly, glia migrate out of these transplants quite actively over relatively long distances. Some of the glia migrate along the same pathway as the axons, but this is not all of them do that. And then we have this single observation with human tissue that are younger. We can't detect glia based on morphological criteria. But still the axons grow perfectly well. There is no difference. So we don't know if glia is important. Should there be glial cells in these mixtures? With the ESL transplants you were here about this afternoon, there are no glial cells generated.
Could I sneak in another question? Would there be an ethical reason against putting in non-human primate fetal material?
I don't know how to justify it. I'm not sure how the regulatory agencies will react. I know that pig tissue, which has been a couple of decades, in the 80s, 90s, there was a lot of interest in using pig tissue for transplantation, also heart and kidney and so on. But then came these retrovirus contaminations and the parvoviruses, and pig tissue got a bad reputation. And the research teams that had worked with it, they abandoned this approach. So I think that, is it called zoonosis, this thing where you carry over viruses from one species to another? remains a concern. There are examples of zoonosis. Of course, the HIV virus is probably an example of that. So I think that large-scale transplantation from animals to humans would be something seen as very risky. To do single cases probably doesn't necessarily make much of a difference. But
also, the human mothers can give consent. So one could argue that it's more ethical to take human embryos than to take primate embryos.
yes.
The numbers of embryos available.
Yeah. Now, I think one should nevertheless just get away from using fetal tissue in the long run. So.
Okay, back to the transplant. So my
Yeah.
question aims exactly at the other neurons, the serotonergic and the glutamatergic neurons. And now that you had the chance to examine the transplants physiologically at different times after transplantation, would they become also more, or would their number gradually decrease because they are in an inappropriate place? Could you comment in the different species, samples?
Yeah, you mean the idea that other neuron types may gradually die off or something?
Exactly.
Or be eliminated? I don't think we have any idea of that, because for human cases, of course, we have simply too little material. For rodent cases, I must confess that there has been so much focus on the dopamine neurons, what they do, the subtype, where they grow, how they function, that we are more than happy to forget about the other things that are. And you could have phrased the question in a different way, which is common. How bad is it to have these other cells? Are they creating problems? Could it be one reason why some patients are not doing so well? I think this is much better addressed in stem cell-derived trials, because the stem cell protocols, in theory at least, can allow for standardization and more precise manipulation by, for example, cell sorting. We don't know, for example, if a 100% pure dopamine neuroblast population would do well.
Yeah.
My guess is that they won't. I think they need partners. Perhaps they need glia, but they might also need other neurons to support themselves in their survival and differentiation and function. But I don't think we have had the material and the kind of experimental manipulations that would allow us to address that well until now. I think now one can do it.
If we can thank Anders again.